ሰላም ሰላም ተመልካቾች ዛሬ ከአሁን በፊት አንድ በፎቶሲንቴሲስ በሚመለከት ጉዳይ አንድ ቪዲዮ ለቅቄ ነበር። ኔክስት ደግሞ ክፍል ሁለቱን እንዴት ነው how can we detect chlorophyll content and photosynthesis plant moisture stress እንዴት እናጠናለን፣ የካርቦን ዳይኦክሳይድ አውት ኢንሌቱን፣ የኦክሲጅን ትራንስፈር ፊውዥን እነዚህን በሚመለከት አንዳንድ መሳሪያዎች ላይ እንዴት ነው ማጥናት የሚቻለው የሚለውን ክፍል ሁለት ቪዲዮ ይዤ ቀርብ ያለሁኝ። ዜን በሳይንሱ ዓለማችን የት እንደደረሰ አግሪካልቸራል ፕሮዳክቱ ላይ ሰብሎችን እንዴት ፕላንት ሞይስቸር ስትረሳቸውን እንዴት ማጥናት እንደሚቻል፣ ክሎሮፊል ኮንተንት እንዴት ማጥናት እንደሚቻል የሚያስችሉ ትልልቅ ሪሰርቾች እየተሰሩ ነው። ስለዚህ እነሱን መሰረት በማድረግ እኔም ያለኝን ነገር ይዤ ቀርባለሁኝ። አብራችሁን ነው።
One or two of them was still holding one. And this is most common when the plant has not been dark adapted for long enough. Some things in the plant relax after 30 minutes. Some things in the plant relax after 3 hours or maybe 6 hours. It really depends on the plant and the behavior. And some things in the plant relax after a few seconds. So the assumption in a dark adapted measurement is that all reaction centers are open and photochemistry is not engaged, and any light that comes back is due to all the reaction centers being closed and then it being able to be fully dark adapted. Then in a light adapted measurement, we need to make sure that the reaction centers are all completely filled so that photochemistry is completely stopped. It's the opposite problem. So in the dark, if we assume that all the reaction centers are open, but if they're not, then we are going to incorrectly calculate our parameters. If we in the light, we think that we are filling all the reaction centers and we aren't, then again, our photochemistry and our assumptions are not correct. So in the dark, we need to assume that all reaction centers are open. In the light, we need to be sure that all reaction centers are fully closed. And so one of the things that you might be thinking about is, well, that's interesting, but what do I want to do with gas exchange? What do I want to do with fluorescence? Well, one of the things that we can do, based on the fact that we assume that all reaction centers are open in a dark adapted measurement and all reaction centers are completely closed in a light adapted measurement, is we can start to look at the efficiency and the effectiveness of fluorescence. FV over FM, this dark adapted measurement, is a very common measurement of fluorescence efficiency. And you can kind of think of it as the potential energy of that leaf. So if everything that was able to go to photochemistry did go to photochemistry, how much of the electrons would be used? And this is a highly conserved variable. In the terrestrial plant kingdom, it is almost always between about 0.78 and about 0.82. It is very highly conserved. And you can think of that as, so 80% of the electrons that were put into that leaf could be going to photochemistry. Where do the other 20% go? They go to that housekeeping that I mentioned earlier. There's always a little bit of electrons, a little bit of energy that's required just to keep the cellular processes running. So not everything can go to photochemistry. Instead, we hope for about 80% to go. And if we see that a fully dark adapted leaf has a FV over FM that is lower than 80%, it usually means that there's a stress response. So the plant is just not as efficient as doing what it was going to be doing because it is stressed. And so it's a very common parameter to check FV over FM to look at heat stress, cold stress, water stress, nutrient stress. All of these things can give an FV over FM response. It does depend on the plant. Some plants are more or less sensitive. Some plants, you take away their water for 1 day and the FV over FM drops. Other plants could be practically falling over at Turgor loss point, and the FV over FM wouldn't change very much. So if you're thinking about doing stress work, which a lot of agricultural research does, because we want to understand what are the stresses that are affecting crop yields, then FV over FM can be a really good parameter to look at. In the light, we can look at phi PS2, and we can think of this as the kinetic energy of the plant. The FV over FM is how much could be going to photochemistry and photosynthesis. Phi PS2 is how much actually is. And that is always lower than FV over FM. And as the light gets higher, in some ways the plant gets more efficient, but in other ways it gets less efficient because it may get to the point that the light is so high that it's starting to stress the plant. We might get into the light inhibiting zone of things. And photo inhibition, light inhibition is a big, huge subject that has a lot of parts, but generally it just means maybe there's just too much stress on the plant from the light related to heat and UV and various other things. So we can look at FV over FM and phi PS2. The next most common thing that people look at is ETR, or electron transport rate. Non-photochemical quenching and QL are less common than that, but generally you will see that people will talk about FV over FM for dark adapted, phi PS2 for light adapted. So, because these are so important and because they're so useful to us, if our assumptions are not true, if our leaf is not fully dark adapted, then our FV over FM will be lower. And then we have to determine, is it lower because this plant is stressed, or is it lower because this plant is not fully dark adapted? In the light, our problem is whether the reaction centers are filled, whether we absolutely 100% fill the reaction centers and therefore have light bouncing off that we can measure in fluorescence. If we assume that all the reaction centers are filled and the light that we get back is because of all those reaction centers being filled, but actually there's some reaction centers over there that aren't filled, then we underestimate what FM prime would be. And then we get into what is called subsaturating flash intensities. And what ultimately happens is that we don't really understand what's happening in the leaf. We think that we have maximum fluorescence, but it's not actually maximal. And Mark Graff and Barry in 1990 were the ones who really started to help the fluorescence community start to look into this. What they did was give a series of saturating flashes, a series of light levels, and they looked at the fluorescence that came out. And they determined that, especially in highlight adapted field-grown crop plants, there was the characteristic that not all the reaction centers were actually being closed, that no matter how much light they gave, there was more response, meaning that there was still more reaction center capacity there. So they started looking at using what was called a flash train to give different light levels at different, and basically to ramp that light to say, well, what if I didn't just look at the FM prime, at the light levels I was able to give, but I looked at the light levels at infinity? What if I determined that that's how much light level I'm able to provide, but what if there was more? And they regressed that FM prime to infinity. This works very well, but it takes a long time. The multi-phase flash technique. And what we do in the multi-phase flash technique is we do an intensity ramp within the traditional flash. So a traditional rectangular flash takes about a second. A multi-phase flash also takes about a second, as opposed to 20 minutes for a flash train. This allows us to get the information that we had from the flash train in a much faster period of time. This is used in light-adapted plants only. Multi-phase flash should not be used in dark-adapted plants because the physiology and the way it all works is such that we won't get good data out of the multi-phase flash in a dark-adapted plant. The technique is not designed for them and it does not work well. You can see, if you would like to look at the paper, that it was written by Lurio et al., that is Shannon, and this looks at how to estimate FM prime. If we underestimate it, then we underestimate everything else. So QE, GM, which is mesophyll conductance, J, which is electron transport, phi PS2, NPQ, non-photochemical quenching. And then if we want to look at J versus A gross, which we get from gas exchange, everything is underestimated, everything is wrong. And we don't necessarily want to add more uncertainty to the scientific process. There's enough of it already. So using the MPF technique means that we get a better idea of what FM prime would be if we had infinite light available. We have a very bright fluorometer on the LI6800, but it is not infinite. So we just want to make sure that we're not underestimating FM prime. One thing to keep in mind in terms of looking at fluorescence data, and this is true of every fluorometer, not just the multi-phase flash fluorometer, is that fluorometers are not calibrated to any particular standard. Obviously, we calibrate them to our standards when we make them, but they're not calibrated to any particular international certification or standard or anything like that. The useful parameters are almost always ratios, and so you need to focus more on the ratios than the individual numbers in most cases. And some parameters do require dark and light adapted or acclimated measurements. And you'd want to take that on the same leaf in most cases. And then go over there and do the 5PS2 on this leaf. That's not necessarily going to work well for you because they're not the same situation. We are comparing two different things, two different leaves, two different organisms. The other thing to keep in mind is that there are some default values. There are default values for light absorption at the wavelengths used, that's alpha. There are default values for dark respiration, and there are default values for the PS2, PS1 ratio. The default values in the software are pretty good, but they are defaults. And if you were able to modify them to reflect what was actually happening in your plant, then your data would be even more specific to your particular measurement. So that's something to keep in mind. I do have some handy references for you fluorescence. They're not written in sometimes scientific speak is just not easy to understand, but these papers are pretty good in terms of explaining how they all. And then there's also a paper that you might want to look at if you were interested in looking at applications. So also we will be providing these slides, so don't worry about madly writing down these references. But these are some references that if you've never thought about fluorescence and you've never thought about using fluorescence, and now you are, then here are some papers that you might want to try looking at, just to give you a good overview of what's going on. About chlorophyll fluorescence. I don't see any questions in the chat either. So we will go ahead and we will go forward. But if anybody does have any questions, please feel free to put up a hand or put it in the chat or something like that. So now we talked about chlorophyll fluorescence. Let's talk about the other part of photochemistry that we are able to measure with the LI 6800. Let's talk about gas exchange. Specifically, let's talk about how the equipment works and what the information that we get is from. You'll notice that this particular presentation, I talk about not only the theory, but I also talk about the mechanics and the electronics, because I want you to understand